about human microbiome, the next frontier of precision medicine. I request the chairpersons to introduce Dr. Dhakefalkar.
Good afternoon, everybody. It's my privilege to introduce Dr. Practice-Dr.Dhakefalkar. He's a microbiologist by training, and he has obtained PhD in genetics in antibiotic resistance in bacteria. AMR has been one of the most challenging situations globally. He pursued postdoctoral research in molecular and cellular immunology. At present, he is the director of Dr. Agarkar Research Institute, Pune. He has more than 110 papers in international journals to his credit and has 10 Indian and US patents to his credit. Sir, we are really privileged to have you here. And he'll be talking on human microbiome, the next frontier in precision medicine.
Dr. Sanjay Gupte sir had told that this is the latest one and very exciting branch which is going to change the whole scenario, he said. So all of us are looking forward to listening to you, sir.
Thank you so much, ma'am, for adding additional monovagganic dabaw on me because of this last comment. At the outset, I must thank the organizers, especially Dr. Gupte sir, for inviting me. I'm an academician, so I will be talking mostly in terms of the laboratory data that has been generated at various places. Since I'm a microbiologist, my interest has been investigating the microbial diversity associated with extreme pristine and other habitats. And whenever I have been investigating such unique sites where the temperature has been more than 100 degrees Celsius or pressure exceeding 2000 psi, or even the highly alkaline or acidic or highly cold environment, you tend to come across various microorganisms. But having said that, the most unique system where you get to see the extensive diversity is the one which is associated with human, and that is something which has been of our interest, and that is something that we have been investigating. And I would like to throw some light on some of the latest things that have been happening in this particular area. Whenever we talk about human and we talk about our anatomy, physiology that doctors are very good at, we must appreciate the fact that we are not alone. And there are some numbers that I would like to share with you, that in 2008 the entire human genome was sequenced, and after that more than 20,000 human genomes have been sequenced, and there are several million exons of human genome that have been sequenced. And all of us have come to one conclusion, and that conclusion is there are more than 20,000 genes in a human genome. That means more than 20,000 proteins that are possibly encoded by our genome. When we compare this data just with the microorganisms which are present in our body or on our body, then that number is staggering. If at all we just try to count them in number, quantify them, then we have 10 to 100 trillion microorganisms, and the genes that they encode are more than 3 million. So if we compare with 20,000 genes by human genome and with 3 million or more than that by microorganisms, then we must appreciate the fact that the proteins that are encoded by these bacteria, viruses, fungi, archaea, and several other groups of microorganisms present in our body have to play some important role in our health, in our being well. So what exactly is this microbiome and how exactly they play an important role? So when we talk about microbiome, we don't restrict necessarily to bacteria. We talk about archaea, we talk about fungi, we talk about various viruses which are present. So it's not only these mere organisms, but it is their metabolites, it is their proteins, it is their genes, their structural elements such as protein, peptides, lipids, polysaccharides, as well as various metabolites such as short chain fatty acids, organic acid, and several other, which contribute, which determine the health and metabolism of any human being. Meaning that there is a theater of microbial activity that actually influences our health, our day-to-day well-being. So what exactly does the microbiome have to do with our health? It is something which is very important. If we look at the microbiome, it is intimately linked with the human health, but this has been done through several activities that they carry out. One of the most important activity has been digestion. And why do I say that? Because in our day-to-day routine diet, we have polysaccharides, we have complex carbohydrates, we have proteins, we have lipid, oily, and none of these components are taken up by our intestinal epithelium or gut as it is. So it has to be broken down. Our body definitely produces several enzymes which help in its degradation, but we must appreciate the fact that major degradation or major enabling force behind degrading these complex nutrients into simplified absorbable element is played by microorganisms who produce a series of unique enzymes which will degrade it. Not only that, these organisms, through their metabolic activities and production of metabolites such as short chain fatty acids, they modulate the immune response also. And this is something which is of very interest to us, that the immune response needs to be regulated. It needs to be maintained in a state of homeostasis. And if at all it has to be maintained in a state of homeostasis, that means there has to be a balance between pro and anti-inflammatory responses. And for that, one of the major component that plays a very important role is regulatory T cells or Tregs. And these Tregs are very fluently controlled by the metabolism of the gut microbiome, and that is something which is again very important. Third, and one of the major important properties or important facets of this microorganism is the protection against the infectious diseases. We eat, we drink many times the street food, the contaminated water, and many times nothing happens to us. There is a great story that when Robert Koch put forth his postulates and when he proposed that many of the microorganisms are essentially causing disease, and he talked about malarial parasite, he talks about salmonella, and there was one gentleman, he just picked up his culture and he drank it, and he said, show me whether I get that disease, and he did not get the disease. And simple reason for that is because he had such extensive, he was probably blessed with a very healthy microbial flora which offered protection against them. And this is something which is very important for us to appreciate, that the terrific lining that is there on the gut epithelium, which is contributed by these microorganisms, is what protects us. These microorganisms not only prevent the invasion by the pathogens, but it also sometimes produce antimicrobial agents which will inhibit the growth and activity of these pathogens, and that is something that our body requires. So several activities such as metabolic health, again very important parameter, because these microorganisms through their ability to produce butyric acid can control the sensitivity to insulin and can control the levels of the glucose, and indirectly or directly it is also monitored by the microbial flora, something that we rarely appreciate. And these are various facts and facets of this microbial population that has been associated with the gut epithelium that really has attracted attention in recent years, especially with the advent of some of the interesting techniques which have become available in the various laboratories. It has become very easy for us to identify what type of microorganisms are present in our body and what role they could be playing. Conventionally, whenever we have to map what type of microorganisms are present, usually the techniques that are used routinely, conventionally in pathology labs are microscopic techniques or which are culture dependent techniques. These are the two types of techniques which are followed. If at all there are microorganisms which are very unique in their morphology, such as spirochetes, malarial parasites, or some of the amoeba or such kind of organisms, yes, they can be identified using the microscopic techniques when you are talking about or when we have considering the specific tissue part. But majority of the organisms, when you have thousands of species which are present in our gut, if you want to characterize them, it probably will not be possible for us to use such techniques. Even the conventional cultural techniques, even if you give the stool sample for analysis, normally the test or media that would be used would be specific for Salmonella, Shigella and such organisms, and you would detect the presence or absence of such organism. What about Clostridium difficile? How many of the labs would look for that? And very few. The anaerobic cultivation of the anaerobic, obligate anaerobic cultures is not very easy, not routinely followed. And that is the reason why there has to be an alternate approach wherein we can not only identify what type of organisms are present, but whether they are significant in the given context or not, that needs to be identified. And for that purpose, the DNA based techniques are very important. And just now we have heard from Dr. Preeti Arora that what type of techniques are available in Green Array Laboratory. I must say that with the availability of the next generation sequencing techniques, it's not really very difficult, and not only it's not even very expensive to carry out the sequencing of the entire metagenome. Now, by doing that, whatever different types of organisms are present, we will get the idea about that. But it is not only enough to know what types of organisms are present, whether those genes are expressed or not, which is the second step. And for that purpose, one needs to look for the transcriptome analysis, that whatever genes are there, whether they are getting expressed into RNA or not. Even if the RNA is there, sometimes that's not enough. That RNA, whether it is getting translated into protein or not. So proteome sequencing is the third component. And even if the proteins are there, whether the substrate is available, then only those enzymes will convert that substrate into a product, and whether that product is harmful or not. And for that purpose, the metabolomics needs to be analyzed, meaning that one has to have a combination of genomics, transcriptomics, proteomics, and metabolomics. So for that purpose, one needs to have access to techniques such as next generation sequencer for genomics and transcriptomics, LCMS or GCMS for the identification of what types of proteins are present, and metabolites which are present. Sometimes, if at all you are going to have a targeted approach, then for that purpose you need to have the microarray. These were the techniques maybe 10-15 years back, they were unheard of. Only the few very sophisticated laboratories would have access to these, and these days this is entirely changed, and many laboratories have it, and we are blessed in Pune to have such laboratory where we can have that. How did we get this idea? How do we combine all this information together? It is not a job for one laboratory or one person. It is a very expensive exercise which needs to be carried out, and this exercise was initially initiated after 2008 by a United States government. It was initiated by National Institute of Health, and the special grant worth more than $250 million was given by White House through their specific funds that were available at their disposal. Why am I telling you this? Because this underscores the importance of this particular area, which was recognized by even the US government. And after that, they came up with the project, and this project was known as the Human Microbiome Project. This was initiated after Human Genome Project was completed. And in parallel, there was another project which was Human Intestinal Tract Program, which was run by the European Union. Obviously, the Americans were far ahead, and they did actually sequencing of more than 3,000 reference bacterial genomes from genomes, and then they investigated, they sequenced, they identified what were different communities, what were different metabolites which were produced. And later on, in the first phase, after identifying all this information, in the second phase they targeted three particular conditions. They used the multiomics microbiome analysis, A, with respect to pregnant women, B, with respect to inflammatory bowel disease, and 3, with respect to type 2 diabetes. So these are the three major diseases where there is a huge body of literature which is available, which has explained the relevance and importance of the microorganisms. So in the phase 1, when the scientist team, they carried out diagnosis, they targeted the intestinal system. That was the most important one, most diverse one. But in addition to that, they also looked for the respiratory tract, they also looked for the vaginal tract, they also looked for the kidneys, and they also looked for the other parts, that is the oral part, where they investigated the microbial population which was there. So when they investigated, they found out specific types of microorganisms which were present. Mainly, in most of the cases, the microbes that influenced the health in particular cases was the one which were different bacteria, and these bacteria were typed, they were identified, they were proposed, and I have just listed. I will not go into details of that because it was very clear that whenever you are looking for the dental plaques, there were anaerobic cultures which were prevalent, and there were facultative cultures which were prevalent when there was a buccal mucosa which was investigated, or even the microbial flora associated with the tongue was investigated. They also did the investigation in certain other cases such as skin, which was the largest organ in the human body. They also studied the urinary microbiome, and they found out that whenever you are investigating the microbial community of the vaginal flora of the urinary tract and the gut microbial population, there were a lots of overlaps, and based on that they identified certain group of microorganisms. Very interesting numbers, that in stomach there are hardly any microorganisms which are present because of the severe acidity, of course, and these contribute to 10 to the power 1 to 10 to the power 3 CFU, that is coliforming units per ml. This number goes up to 10 to the power 6 in duodenum, more or less the same in the jejunum and ileum, but in colon the numbers goes up to 10 to the power 10 to 11. That means there is a thick paste of microorganisms which is present in such cases, and these microorganisms, if at all their balance is damaged, their balance is disturbed, if at all there is dysbiosis which is known, then it would cause a lot of differences in the gut brain axis, the gut brain endocrine axis, gut heart axis, and several other such parameters are there. In such cases, everywhere there are adverse effect. And what happens when there is a eubiosis? Everything is fine. At that time, if you look at the metabolites, then the short chain fatty acids, the antioxidant molecules, then there are the healthy gut epithelium, and there are several other such parameters. You will see all these molecules, all these genes expression is enhanced. And as compared to that, if you look at some of the other parameters such as inflammatory mediators or pathogenic organisms, their expression is somehow decreased or it is decreased. Now, what does it tell us? This tells us that these microorganisms play a very, very important role. And if at all you look at some of these the neurological disorders, I'll just take a minute to yeah. So in case of Alzheimer's disease, in case of Parkinson's disease, in case of autism spectrum disorders, in case of gastric cancers, you will see that in all these cases, the studies have identified what different types of bacteria are present, and uh, there what exactly they changes. I mean, whether there is an increase in number of these bacteria or decrease in their number of bacteria has been proven. So it actually gives us some kind of insight with respect to the bacterial association with these diseases, that in cases of such kind of conditions, whether you are going to get upregulation or down regulations in this particular population. And that is something that is of our interest, because then it gives us idea whether we can use these tools as even a diagnostic tools, and that is something which has been of interest. So what does this all lead to? This all leads to a path towards precision medicine. And why do I say that? It is because when you take a look at all these factors, that is the multi-omics approach, where you have investigated the genomics, where you have investigated the proteomics, the transcriptomics, the metabolomics, and also taken into consideration the individual human genome, then that gives you a specific interaction between the body, between the individual and its microbial flora. Once you have understood that, then that can lead to a targeted approach. I'll give a very simple example. Let's imagine a combination of prebiotic and probiotic. I'm sure all of us have heard this terminology. You have lactobacilli, bifidobacteria, all such organisms which are known to be probiotics. And on the other hand, you have also heard of prebiotics such as fructooligosaccharides, inulin, and several other such dietary fibers are there. Now, how can we combine these together as a personalized or precise medicine? You can carry out the sequencing of the microbial population. When you sequence the entire metagenome, from that it is very easy for us to identify which gene belongs to which organism. You can, in fact, by using the technique called as binning, you can separate this individual sequence of individual organisms. From that, we get a clear-cut idea which organism has ability to use or degrade which dietary fibers. So from that, we essentially get the idea about the choice of those dietary fibers which are not used by harmful organism, but which are used by only the helpful bacteria or friendly bacteria. And you have the combination of those probiotic cultures which are required. For example, you want high butyrate producing bacterial culture, or you want high acetate producing bacterial culture, then you can combine them together. And with this approach, for a specific individual, you can design a targeted one. A first fecal transplant case that happened also can be considered as one of the interesting parameter. We all must have heard of this particular case where it happened in United States when a person was suffering from bacillary dysentery for a very long time, and he was not responding to any of the 28 different antibiotics that were given to him, either as individuals or in combinations. And at that time, doctors decided to try the fecal transplant when his wife, who was healthy, this particular individual had lost more than 40 kgs of his weight, and he had become absolutely fragile, bedridden in hospital. And at that time, since his wife has been healthy for a very long time, since they both shared the same environment, same dietary preferences, they took the fecal material from that, and through the nasopharyngeal route, they introduced it into his body. And when it was transplanted, within a very short period of time, that is within 6 weeks time, this person gained 15 kgs of body weight. And that was something which was a very unique case. Again, it was individual at that time. Whenever these doctors had to go ahead, they had initially collected the sample from this individual, investigated the microbial flora, understood what type of microorganisms he needed but were not there. They investigated the microbial flora of his wife. They investigated and confirmed that there were no pathogenic organisms were present. They confirmed the compatibility, and then this was used. And it was all possible because of the availability of these latest genetic techniques or analytical techniques. And that is how people go ahead and do that. So we all know that we cannot change our genome, we cannot change whatever is our genetic makeup, but we know that microbiome can be influenced. It is very easily influenced by dietary preferences, it is very easily influenced by the exercise, and even by diseases, and even by the drugs. We know it can be manipulated. We can introduce certain bacterial cultures from outside. So what it tells us is, as per our requirement, whenever we want to do that, and when we anticipate that such kind of bacterial introduction is going to be useful, we carry out the genome analysis. We use this particular sophisticated technique to analyze, and then we can prepare a customized formulation and introduce this genome into body, and then it can be very easily used. There are several examples, especially with respect to type 2 diabetes, that when there is abundance of butyrate producing bacteria, it is linked very strongly with type 2 diabetes. With colorectal cancer, there is a very strong correlation between prevalence of the fusobacterium and porphyromonas, which is a dental pathogen, by the way, and it has impact on the colorectal cancer. There is a liver cirrhosis, where there is a low abundance of friendly bifidobacterium and lactobacilli, and there is a higher abundance of potentially harmful bacteria such as members of Enterobacteriaceae and Valloneriaceae. So these are the several cases where we know. So from this kind of data, we can actually look for a combination treatment to inhibit the harmful bacteria and to promote the friendly bacteria. And if at all we come up with such kind of combinations, then that would definitely help us in making use of microbiome for the application in the medicine. And there are several examples of such therapeutics, which are dietary interventions, which are absolutely fine. And there are prebiotics, probiotics, synbiotic, which is combination of both. There are antibiotics, there are fecal microbiota transplantation, and now these days you get the capsules, and it has become very acceptable way of taking the fecal transplant, and the company will make the customized capsule for consumption consumption and one can use it. There is a customized phage therapy and there is a customized biotherapeutics where certain antibodies which are targeting certain harmful proteins or agents, when they are to be targeted, these antibodies are expressed on the surface of bacteria, and then those bacteria are introduced so that they will get into our environment and they will express such kind of antibodies. So there are different ways in which we can use them, and this in this particular slide I have tried to summarize. I will not go into the details of that because in CRUSC we have discussed most of these part, but I would just like to differentiate at this particular point what exactly is the difference between the probiotic and fecal transplant. Normally, if at all we want to see an individual where the host physiological augmentation is to be obtained or achieved, for that purpose we can give the probiotic treatment. When we know that because of the presence of pathogen, diets, antibodies, alcohol, and environmental parameters and several combinations of one or few or more or all of these, then there is a disturbance of the microbial community structure which is known by the term dysbiosis. Then obviously we have to have some kind of intervention, and at that time to replenish the entire gut flora with the healthy flora, normally a combination of broad spectrum antibiotic followed by the treatment with the microbial population is followed to achieve the microbial equilibrium and restoration of the healthy flora. And this is where the fecal transplant from with the availability of the healthy donor who has the same dietary preferences, who also has the which is who also is from the same environment, can be used, and this is something that can be achievable. So towards the end I would just talk mainly about the examples of the microbiome based therapeutics. I have listed certain examples for just for our information, and these are some things which have been investigated, which have been proved to be useful. There are certain cases where the challenges and ethics about the use of this microbiome needs to be discussed, and there are challenges in the form of suitability of the cultures that we are using, the biosafety of the cultures that are to be used, especially some of the borderline cases where you are using streptomyces species. There are certain saprophytes, certain pathogenic and some friendly species. You have to have a very accurate identification as well as quality check to use such kind of cultures. You have to have a very terrific and in-depth microbial characterization that has to be done, and after only having the several human trials to prove its stability, to prove its usefulness, to prove its harmlessness, then only we can go ahead with this. So there are challenges which we have listed, some of them. The unidentified microorganisms, especially the viruses, because whenever we are doing this, these are some of the parameters which normally are not very well understood or investigated. Some of these cultures need to be properly identified. That is one part. Understanding the interactions and dynamics and adaptation is something that needs to be investigated, and identifying specific microbial molecules as well as the mechanisms which regulate the host health are something which has to be identified. I would say that at this particular time we are in early days. We are in early days because this is a new up and coming branch of science. This is something which is going to have profound implications on our health. This is something which offers as a wonder pill which would have ability to cure several diseases. But if at all it was the case, then it would have done so earlier. So there is need for a lot of investigation that we need to carry out, and this is something that has been done. As in the previous presentation, Dr. Arora mentioned that they are collecting the data for the human population, for the Indian population. This is something which is very important, and I must stress on this, that there are several efforts, but there is still a paucity of information regarding this. And if at all we want to take this further, and if at all we want to find these as a solution for some of the diseases, which can be affordable solution, which can be in the interest of society to achieve such societal benefits, I believe that there is a need to work very closely. I always believed that doctors in India are where they are the best of the brains. But maybe because of several reasons, as every individual doctor has to look for so many patients, very few, if any, doctors are involved in the research. But that is the need of the hour. If at all, even 1% of the doctor, even any one of any or few of the doctors are ready to give 1% of their time for research related to development of the newer therapies, which could be for useful for the Indian population, then I believe that our country will have a great future. Again, I would like to thank the organizers for giving me opportunity to share some of my thoughts and introduce this interesting topic. And I hope that when we meet next, we will have far more data to discuss. Thank you. Thank you so much.
Thank you, Dr. Prashant, sir. It was a wonderful and really a talk which has given a lot of vision to every listener present in this house. And yeah, thank you for introducing most of the doctors to the new horizon in the field of medicine and the future, which is not very far off. It has to come soon. So thank you so much.
Thank you, sir.
Can we take up our questions, sir? Yeah.
How do you do finger transfer?
Okay. So now this has become a very routine technique where there are certain companies which offer you these services. So what doctors do is they initially carry out the screening of the individual, the donor, and they also carry out the screening of the recipient. They identify the need of specific type of cultures. And then in those cases, those cultures are put in a capsule in a lyophilized form, and these capsules are administered. Earlier days, they used to prepare the enriched solution of bacterial culture, and through nasopharyngeal route, they used to introduce this. Now they don't do that, they just prepare the capsules, and those capsules can be engulfed.
Dhakefalkar sir, yeah, that was an excellent lecture and giving future insights. I would say, Madam mentioned about barcoded babies. Probably you are speaking of barcoded poo pills in future. So I request Dr. Gupte sir, Dr. Sanjay Patil sir, and President Dr. Varyani sir to join the felicitation of the speaker and the chairpersons to join. I request Gupte sir to felicitate the chairpersons. Padma Iyer madam, she is trustee IMA Pune. Dr. Alka Shirsagar madam, treasurer IMA Pune. Dr. Vaijayanti Patwadan madam, managing committee member and past president IMA Pune. We thank all the chairpersons for chairing this session.
Thank you, the chairpersons, for chairing the session. I take this opportunity on behalf of IMA Pune, from bottom of our heart. We wish to thank Dr. Sanjay Gupte sir and Dr. Asmita Gupte madam for this wonderful CME and oration. IMA Pune wishes to thank you, and I request Dr. Sanjay Gupte sir. Sanjay Gupte sir, please come on to the dais. I request President Raju Varyani sir, Dr. Sanjay Patil sir, Dr. Navrange sir, and Dr. Meenakshi Deshpande madam to come on to the dais. we wish to felicitate Gupte sir. Madam, will you be okay? We all of you, we request you all to come down and felicitate Madam.
I request Dr. Aarti, please join Dr. Surgeon. Moving on to the next session, we have a lecture by Dr. Seema Gaikwad on earliest and fastest detection of infectious diseases. For this, I would like to invite the chairpersons, Dr. Raju Variani sir and Dr. Sanjeev Khud, an eminent gynecologist, to chair the session.
We also wish to thank the whole team of Dr. Gupte Hospital and especially Mr. Nitin Gupte sir, Mr. Avinash Joshi sir. Please, are they here? Okay, just खाली गेलेत का? नाही, काय असतं, काम करणारे लोक पुढे येतच नाही, ती नेहमी मागूनच असतात. सो मेनी टाइम्स बोथ ऑफ देम केम टू IMA फॉर मीटिंग्स. सो मच वी डिस्कस्ड आणि या सगळ्यातून इतका सुंदर प्रोग्राम आज आपला झाला. त्या सगळ्यांचे खूप खूप धन्यवाद आहेत. कुठे आहेत? ओके ओके सर. सर, थँक्यू व्हेरी मच. Over to you, the chairpersons for the session.
Hello, yeah. Good afternoon, everyone. It's indeed a pleasure to be a part of this wonderful program. And my sincere thanks to Dr. Sanjay, Madam, and my friend Raju and the whole team. I must thank Sanjay for giving me this opportunity, because this has given me an opportunity to meet Madam, the one who gave a fabulous oration. And secondly, Madam taught me how the baby delivers, baby pelvis giun. She is the first one when we were residents, junior residents, to have learned the first lessons of obstetrics from you. And Sanjay is the one who taught me hypertonic, hypertonic saline घेऊन abortions केली जायची. So the first lessons actually were learned from them.